So we're going to get started and begin on the first introduction to T-cell engagers and general mechanisms of action. So I'm going to spend the next few moments, I think, setting the stage for what you're going to hear from our faculty in the coming moments as well. So when we think about immune therapy in the context of multiple myeloma, I think about it in sort of four buckets, if you will. I think there's passive or active antibody-based immune therapy, and the T-cell engagers now fall into this category as well. There's passive immunity targeting a receptor. This is things like CD38, SLAMF7. The indirect way that is listed on this slide really refers to things like PD-1 or LAG3 or TIM3, sort of other targets that may not directly impact the tumor, but indirectly activate immune function to allow us to get anti-tumor immunity overall. There's also the concept of adjuvant therapy, which really focuses on immune boosters. These are theoretically more of the dendritic cell or peptide vaccines. And then finally, active therapy, which I would have argued probably the prototype for this for the longest time was allotransplant. Fortunately, we've moved beyond the era of allotransplant, and now what we're really doing is focusing on CAR T cells, which does have off-target therapy, off-target effects as a potential risks as well. So when we think about options for relapsed and refractory myeloma that really focus on immune areas, the area on the left is probably the one that has had the greatest impact for the longest time in the context of myeloma. These are the immunomodulatory agents, which were thought initially to have pleiotropic effects, multiple mechanisms of action. But what we now know is that their effects are largely through targeting cereblon directly in the myeloma cells, but also by targeting T cells and NK cells. And this ultimately leads to T cell and NK cell activation, which makes a great adjuvant for many of our immune therapy approaches across the board. And so I think the idea of lenalidomide and pomalidomide and the second generation cell mods such as iberdomide and mezigdomide, these formulations I think really do form, in my mind, the ideal partners for many of our immune therapy approaches. Now, if you look at the right side, you'll see the antibodies and the bispecifics. Elo, Dara, and isatuximab are sort of the old boys, if you will, in the armamentarium. And then now we have teclistamab, talquetamab, and elranatamab. These are the new kids on the block that really are bispecifics and work by different mechanisms of action. Finally, we've got the cell therapy approaches, two cell therapy approaches that are approved, and these include ide-cel and cilta-cel. So as we think about mechanism and structure, it's important to recognize that many of these bispecific antibodies have very different structures based on the biology of their simple plan overall. And so if you look at the bottom, these are examples of bispecific antibody structure overall. But in their heart, they really are formed by one part of an antibody that binds CD3, as you can see from panel A above, and the other part that binds a plasma cell. So that plasma cell target can be BCMA, it can be GPRC5D, it can be FcRH5 in the ones that are furthest along in clinical development right now. And so what you functionally do is bring a T cell in close proximity to a malignant tumor cell, and that ultimately results in myeloma cell death. Now, the structure of these can be quite different. This class is often referred to as BiTEs. Just to be clear, BiTEs are actually one type of bispecific and actually use a technology that is unique to drugs like blinatumomab. The bispecific category or descriptor is probably more broad and does fit with with many of the whole class of drugs, as opposed to BiTE is really unique to a product from a single company. So there are differences in that terminology. And what we know about bispecific antibodies and T-cell engagers is that they really have multiple effects. Again, there can be bispecifics that target NK cells as well as plasma cell receptors. And what we know in overall simplicity, if you will, is the idea that these are able to bring together elements of the immune system that have been physically separated in order for the myeloma cell to make themselves somewhat protected, if you will. If you were a cancer cell, the first thing you'd want to do is create a sanctuary where you were free of the peskiness of the immune system. And that's what myeloma cells like to do. T-cell engagers break down that barrier and help us to potentially get in between these cells and bring T-cells in close proximity to the malignant tumor overall. Now, the first that I want to talk a little bit about, and I think, wait, where am I? Did I get lost here? Let's see if this will work. So the first I want to touch on is BCMA, because BCMA probably has the most data of any of the T-cell engagers that we have so far. And as you can see, BCMA does in many ways represent a perfect target in myeloma. It is expressed on the end-stage plasma cell. It is expressed on a few progenitors of B cells that ultimately become plasma cells. And so this is why some of the infection complications that we see in patients who are getting BCMA-directed therapy can be an issue, but recognize that BCMA is an important survival factor for myeloma cells. And so for this reason, it does in many ways represent a unique and really important target overall. When we begin to think about another one, which is GPRC5D, you'll see here, again, GPRC5D is almost uniquely expressed on plasma cells. When you look at malignant cell lines, it's not on any other tumor or cancer. GPRC5D is expressed, however, on some gut epithelium as well as oral mucosa. And so for that reason, it does in fact have some side effects that are different from BCMA. And those are some things that we're going to hear about in today's talks that will help you to anticipate what to get from any given patient. And finally, FcRH5. FcRH5 is expressed on plasma cells with almost 100% prevalence. It is expressed on a few normal B cells, but not to the same extent that BCMA is. So infectious complications seem to be less common with FcRH5 targeted BCMA or targeted bispecifics. And cevostamab is the prototypic agent in this area as we go forward now. So this represents the third big category of bispecifics, and you're going to hear more information about each of these three targets in the coming talks.